Good afternoon, everybody. It's my pleasure to welcome you to another edition of the Bridges Seminar for the Hellenic Initiative Against Alzheimer's Disease. Today, we have an interesting, exciting speaker and a very novel topic, which is the emerging understanding of a new condition that is referred to as late. You will hear all the details from our speaker. So David Wolk is currently professor of neurology at the University of Pennsylvania. He's also co-director of he's also director of the Alzheimer's Disease Research Center, co-director of the Institute on Aging, chief of cognitive neurology, and of the Penn Memory Center. His research has focused on cognitive neuroscience of memory decline associated with aging and Alzheimer's, and with the use of biomarkers, including behavioral and neuroimaging, to differentiate healthy aging from transition to AD. I've known David since he was a medical student at Hopkins. He was one of the stars that we tried to keep here that we did not succeed in keeping here, but he has gone on to have an amazing career at UPenn and is now, I think, one of the world's experts on late. And so I'm delighted, David, that you were able to be with us today. Welcome.
Thanks so much, Kostas. And of course, my time at Hopkins was some of my fondest moments during my career. So definitely miss it. I'm going to share here. So let me pull up my slides, if I can figure that out. Looks like I can. Assuming you're all able to see.
Yep.
So I'm going to try to give kind of a broad overview about what late is and why I think it's particularly important now as we enter into a more sort of therapeutic stage of Alzheimer's disease. I think we're at the very beginnings of that, but I think we've definitely made progress. Here are some disclosures. So just to give you an overview of what I'm going to talk about today, I'm first going to try to frame the discussion of late into the general challenge of dealing with co-pathologies in the context of AD therapeutics. I'm then going to spend a little bit of time defining what late is, and we can talk about the cognitive and imaging profile of late, both when late is kind of the primary driver of symptoms in individuals, as well as when it is a co-pathology with Alzheimer's disease. And finally, I'm going to sort of shift a little bit into how some of the work we're doing in trying to detect late when it is a co-pathology. And then at the very end, I'm going to briefly go through what are clinical criteria for late that we proposed about a year ago at this time. So I think just to set things up, as I think most of you are aware, we are in a new era with disease-modifying medicines for Alzheimer's disease. There are two monoclonal antibodies to amyloid in the brain that actually have remarkable reduction in their ability to clear amyloid from the brain. So these figures reflect amyloid PET measures, something called a centiloid measure. And in the untreated group, you can see that there's very little change in amyloid PET measure over time. But with treatment, there's really remarkable reduction in PET signal of amyloid. And those of you who are aware of these measures, an 80 centiloid drop is quite remarkable for donanemab. And actually, by the time people completed these trials, about a year and a half into it, around 80% or so are actually cleared of amyloid. And so this is one of the patients in our center who started off amyloid positive. After 18 months of anti-amyloid therapy, had an amyloid negative scan that really is indistinguishable from a normal adult scan. And so just taking a step back, it really is a remarkable thing that, one, we can see the pathology of Alzheimer's disease, and two, that we're able to have an effect on that pathology, something we think of as very inert, so dramatically. But of course, none of this matters if it doesn't impact patients' lives. And in fact, these drugs do slow down the progression of Alzheimer's disease modestly, but very statistically significantly, by about a quarter to a third. So not a grand slam, but they do seem to slow down the course of the disease. And so one question is, why, with this much robust amyloid clearance, why isn't it an even bigger effect? And I think there are probably lots of reasons for that. But one thing I do think is worth always kind of keeping in mind when we're providing treatments to individuals with Alzheimer's is it's an incredibly heterogeneous condition. And I think this is really very beautifully illustrated in this figure, which shows a measure of function and cognition called the CDR, Clinical Dementia Rating Scale, sum of boxes, over a 24-month period in patients with Alzheimer's disease. And if you look at change from baseline, it's just a mess of different and variable courses of patients. Yes, on average, everyone is getting worse, but some are getting worse much more rapidly than others. And I think anyone who sees patients with Alzheimer's disease are very aware of the fact that there are many dimensions and likely sources of this heterogeneity. In terms of dimensions, we just showed there's differences in the rate of progression, age of onset obviously varies quite a bit, the nature of the cognitive and behavioral symptoms, and even where the pathology is in the brain will vary across these individuals. And there are probably lots of different reasons why there's this heterogeneity. But one of them, which we're going to kind of hone in on with this talk, or is going to be the primary focus, is the presence of co-pathologies, which are quite common, as I'll show in a second, with Alzheimer's disease. So this is our brain bank at Penn, and these are pathologically confirmed Alzheimer's disease cases. And if we look at different proteinopathies, we see only about a third of patients just have amyloid and plaques and tau tangles, the defining pathologies of Alzheimer's disease, whereas about 2/3 also have alpha-synuclein, and around 2/3 also have TDP-43 pathology. And in fact, a good quarter of patients have amyloid, tau, TDP, and alpha-synuclein. So it's very common to have all these different pathologies. And the one, again, that we're kind of focusing on today is late, which is related to TDP-43, as we'll talk about a bit later. And that, again, makes up at least 40% of the pie of patients with Alzheimer's disease. And in fact, if you include vascular disease, in addition to other proteinopathies, the degree of co-pathology with Alzheimer's disease is even higher. This is a study from the Rush University autopsy study. They classified patients a little different here. These are people who are clinically called Alzheimer's disease. And so the the non-blue shades are associated with misdiagnosis, people who didn't have Alzheimer's. But if you look at pathologically confirmed cases with that are the kind of blue shades, the least common form of Alzheimer's disease is just Alzheimer's disease. So it's actually quite rare to just have Alzheimer's disease. And I think that's important for us to keep in mind. And so when we got to go back to thinking about, well, why didn't we see more of a benefit with these drugs? Well, one possibility is that we're only targeting one of the proteinopathies associated with Alzheimer's disease, in this case, amyloid. And I do think it's really important to try to sit back and think, well, how do we, in an individual patient or overall, figure out how much Alzheimer's disease is driving the cognitive symptoms in patients? One approach on a more broad level that's been taken to look at this is work that was done at Rush University, where they calculated attributable risk in their autopsy cohort. And what they found was that Alzheimer's disease only accounted for less than half of the attributable risk of the symptoms of Alzheimer's, whereas alpha-synuclein, vascular disease, and late here in yellow accounted for significant proportion as well. And so it's perhaps the case that there's some sort of ceiling to how much we can slow down the disease if we're not targeting these other conditions, at least in some cases. Okay, so that brings us to talking more specifically about late. So what is it? It's an acronym. It stands for limbic predominant, age-related TDP-43 encephalopathy. Limbic predominant because that is the main part of the nervous system it affects. Age-related because it tends to occur later in life. TDP-43 because that's the protein that is associated with late. And encephalopathy because they wanted the acronym to spell something and they needed an E, and that was the best choice that they had. And that actually is largely true. So one definitional piece, people often refer to late neuropathic change as the pathology of late versus late itself, which is the kind of clinical manifestation of the disease, kind of akin to when people talk about Alzheimer's disease neuropathic change and then Alzheimer's disease clinical symptoms. TDP-43 is a ribonuclear protein that plays an important role in RNA regulation and DNA regulation. And actually, in general, this protein in normal function can actually shuttle across the nucleus and into the cell body and back again. But in a pathologic state, it tends to aggregate in the cell body, clears the nucleus, and that is seen, as we'll talk about in a minute, in a number of conditions, including FTLD, ALS, and also in late. And it's still quite unclear the degree to which these aggregates reflect a sort of toxic gain of function as the cause of why TDP-43 is problematic, versus it's a loss of function in its RNA and DNA regulatory function. And it can be both of those things as well, and that remains unclear. Currently, there are no molecular biomarkers for TDP-43, although there are a few promising ones, including measures of something called cryptic peptides or cryptic RNA that are associated with misregulation of TDP-43. And actually, Phil Wong at Kostas's shop has done a lot of work in this area. So there's actually kind of a long history to TDP-43's role in late life cognitive decline. Dennis Dixon in the sort of mid-90s actually described something called hippocampal sclerosis, different from the medial temporal lobe sclerosis that we often think of as associated with epilepsy, at least as neurologists. And he found actually in about a quarter of patients over the age of 80 who didn't have Alzheimer's disease had what he called hippocampal sclerosis. And on the bottom here is a histology of that, in which the sort of middle figure, and I don't know if you can see, maybe I'll put in the laser pointer. This middle slide shows sort of normal pyramidal cells within the CA1 subfield. And in hippocampal sclerosis, there's this just severe dropout of pyramidal cells in CA1 and in subiculum in particular. And Dennis noted that these patients had a memory disturbance that seemed disproportionate to deficits in other cognitive domains. And in some ways, that was very prescient of the clinical syndrome that we now think of as associated with late. TDP-43 actually wasn't discovered until 2006 by Manuela Newman and Virginia Lee here at Penn. And it was initially described as being related to FTLD, ALS spectrum conditions, as most of ALS is associated with TDP-43 and about half of FTLD is associated with this proteinopathy. Very shortly after, TDP-43 was found to be quite common in hippocampal sclerosis, more than 90% of those cases. Over the next decade, work at a number of different labs, including at Mayo with Keith Josephs and Julie Schneider at Rush. There was a lot of work showing that TDP-43 and AD seem to have independent effects on hippocampal structure and memory decline with aging, and that it was quite common to see TDP-43 in the medial temporal lobes of older individuals, even in the absence of Alzheimer's disease, with about a quarter of patients over 80 with this condition, but that it was even more common, and I'll show some data on this, in cases with Alzheimer's disease as well. But I think late kind of hit the map as something that people started to really think about a lot more when it was codified into a pathologic definition that was led by Pete Nelson, University of Kentucky, where he defined the condition pathologically. In 2023, there was a update to kind of simplify the pathologic criteria. And if you look at the criteria that Pete mentioned, it required TDP-43 that in an initial stage was in the amygdala. There was then progression to the hippocampus, and then stage 3 was associated with middle frontal gyrus. I should note that both Keith Josephs and Julie Schneider had previously described sort of staging of this late life form of TDP-43 that is distinct from the patterns seen with FTLD in many cases. And I do think their staging scheme is more granular and actually gives us some hints as to what kinds of symptoms and where we might expect to see neurodegeneration. So Keith Josephs, for example, pointed out involvement of insular cortex, and Julie Schneider pointed out the involvement of the anterior temporal lobe as well as orbital frontal cortex as well. Again, looking at the numbers of cases, it is a highly prevalent condition in pathologic series in both age and with Alzheimer's disease. And so if we look at just age in general, we see a steady increase that becomes quite high when people get into their 80s and even 90s. More than 50% when you get into your 90s have stage 1 through 3 of late. Similarly, in the NAC cohort, this is the National Alzheimer's Coordinating Center cohort in the States that is associated with all the Alzheimer's disease research centers. There was also this relationship with age. It may be a little bit more skewed because the population in NAC is it's a convenient sort of memory clinic sample in most cases, whereas in the Rush cohort, it's more of a population-based study, which is always, I think, important to keep in mind when we think about prevalence numbers. However, we also see even more late or a greater proportion of individuals with late when we look in cohorts that involve cases with Alzheimer's pathology. And you can see here, these yellow bars reflect people without late, and the blue bars reflect people with late in a very large sample from 13 community or population-based studies. And again, when you get into later stages of Alzheimer's based on CIRAD or BROC, it's more than 50% of cases. But I think, again, it's worth pointing out in this cohort series that even in cases that would be considered low or no Alzheimer's disease neuropathologic change, about a quarter of patients had late pathology. So why should we care about it clinically? Well, I think one, as we've talked about, there's a growing awareness that it is an important modulator of late life memory decline. And I think that recognition, as is often the case, was kind of amplified by the reification of late as a kind of distinct, at least pathologic entity. With this increased awareness, though, I think clinicians, we tend to look for patients that fit these sort of pathologies once we know about them. And I think there's an increased recognition in the community that we're seeing patients with symptoms and syndromes that are suggestive of late. But I really do think that the importance has been amplified by the emergence of disease-modifying medicines. In particular, late is a mimic of Alzheimer's disease. And now that in many of our patients that we're seeing coming in with memory complaints, we're getting biomarkers for Alzheimer's to determine whether they're potentially reasonable candidates for anti-amyloid therapy. Many of them end up not having Alzheimer's disease. And so then the question is, what do they have? And many will have late. The other, I think, really important point is what I alluded to earlier, which is that late frequently co-occurs with Alzheimer's disease. And we don't know the degree to which that influences outcomes with these therapies. And so I think it's really important when we think about risk benefit of these medicines for us to get a better handle on who has concomitant pathology with late. So to frame what these patients look like, I wanted to present a clinical case. This was a retired engineer in his 80s with forgetfulness that I saw. The family had noted forgetfulness, but it became really salient to them when their grandson made the high school hockey team. That was apparently a real big deal for this family. And the next day, after everyone had been really excited the day before about this, this patient forgot that he had made the team at that point and kind of asked about when the tryouts were. He needed more notes for reminders. He had some inefficiencies with the finances, but otherwise was able to do his instrumental activities of daily living. And therefore, at worst, is probably someone in the mild cognitive impairment stage. And there was no change in personality or language that they picked up on. He had a mini mental of 30 out of 30, but borderline memory performance. And this is his testing. This is a battery that we do across a variety of different cognitive domains. And These are Z-scores, and the more negative you are, the more impaired you are. About 1.5 Z-scores below the mean is reflective of maybe a threshold of impairment. And the important point here is that the tests that he was impaired on were all memory tests, both visual and language or verbal memory measures, whereas other domains were largely spared. And so we thought this patient had mild cognitive impairment with primarily amnestic syndrome. Over time, he actually progressed really, really slowly. So these are 8 years that we followed him. This is MMSE, which did drop, but kind of bounced around. And so even 7 years later, his MMSE was only at 26. So not a lot of decline there. Measures like naming didn't really change much. Fluency measures were relatively intact throughout. Memory, though, this is his 10-item word list. He started off borderline and then kind of bottomed out and stayed that way. So he was quite amnestic throughout his course. By the third year we were following him, he kind of reached a dementia level of impairment, and we diagnosed him with dementia due to Alzheimer's disease. In year 5, he participated in a research study and had an amyloid PET, and lo and behold, the amyloid PET was negative. This was his MRI, and I think it has features that we tend to see with late, which include in the collateral sulcus. It's a little bit hard to see here, but we often see really significant thinning of the gray matter, particularly in transentorhinal cortex into BA35, which is also known as perirhinal cortex. There's flattening of the head of the hippocampus, which is quite atrophied, but there's also involvement of the body, the hippocampus as well. So really small hippocampi in this patient. And he was the first case at our center that we actually clinically diagnosed with late based on what we saw here. And lo and behold, he had late pathologically. These are in sort of brown, are aggregates of TDP-43. He had late stage 3, which is the most severe form of late, with hippocampal sclerosis and kind of very low level Alzheimer's disease. He had in the ABC scoring, a BROC 1 or a B score of 1, which is BROC 1 to 2. And so very minimal Alzheimer's disease at that point. So this case, I think, illustrates what has been seen now in a number of studies, mostly based on autopsy studies where people look back at antemortem data, in that episodic memory in these cases tends to be a relatively isolated area of impairment in cases with late, whereas in Alzheimer's disease, we see a more typical amnestic multi-domain syndrome where other domains are involved as well. Another feature of this case, and that's typical of late, is that if we look at a kind of global measure of cognition over time prior to death, cases of Alzheimer's disease have a continuing decline over time. Late tends to be slower, though, than Alzheimer's disease. And so in some ways, it's a much more indolent condition than Alzheimer's. In general, again, episodic memory is a relatively isolated domain involved, although we and others, and I'll show a little bit more data in a second, have also seen that semantic memory also may be involved to a lesser extent in these cases, in part likely due to the fact that there's this spread of pathology into anterior temporal polar regions that are more associated with our semantic networks. In general, there's an argument out there that there's not really significant neurobehavioral associations with these cases, and I agree that these cases don't look like FTLD, but I think we're seeing more and more that there are more subtle behavioral symptoms, perhaps some degree of disinhibition and apathy, and actually have some work here that's in progress showing that may be the case relative to Alzheimer's disease alone. So one thing I have mentioned a couple times now is hippocampal sclerosis and how that's associated with late. And what's that relationship? Well, it turns out hippocampal sclerosis probably makes up about a third of cases with late. And I think you could think of hippocampal sclerosis in general as a kind of more severe form of late. About 2/3 of late cases, though, don't have hippocampal sclerosis. And then another important point is with hippocampal sclerosis, late probably makes up around 90% of those cases, but there are about 10% who have other causes. Vascular is another cause. There's some other less common proteinopathies that can be associated with hippocampal sclerosis. When we look, though, at cognitive impairment prior to death across domains in these different conditions, again, Alzheimer's, we see an amnestic multi-domain syndrome. Memory is the most affected, but relatively early on, all of these other domains, semantic memory, working memory, perceptual speed, all kind of go below a threshold relatively early in the course as well. TDP-43 without hippocampal sclerosis tends to be more mild, but to mostly just affect memory. And when patients have hippocampal sclerosis, they tend to have more severe atrophy, obviously within the hippocampus, but also in later stages or later areas that we associate it with late, including kind of more temporal polar regions. And we see not just more severe memory impairment, but also semantic impairment in these cases, as well as in some cases as they get later, more kind of executive function, speed of processing changes. Again, when we think about where we expect to see neurodegeneration and symptoms, in the case of TDP-43, it really seems localized to where TDP-43 is. And so this is a post-mortem study done at Rush looking at volumetrics and association of degree of atrophy. Hotter colors are more atrophy. And basically you see severe medial temporal atrophy, but also involvement into temporal pole and orbital frontal cortex. This also has been seen in antemortem imaging of the whole brain in cases with late, in which there's kind of anterior medial temporal and temporal polar involvement to a greater extent, some degree of frontoinsular involvement as well. But one of the things that we see, again, that I've mentioned already, is really severe hippocampal atrophy in these cases with late. And actually, this is data from Rush. It's a post-mortem study looking at hippocampal volumes. And if you isolate on cases with AD alone or late alone, you see that, if anything, late has a much more significant degree of hippocampal atrophy than Alzheimer's disease does. I think clinically, one can use visual rating scales like the Shelton scale that Phil Shelton's had developed. Patients with late, or at least patients that that we're more sure of that have late, tend to have stage 3 or 4. They tend to have really significant hippocampal atrophy, particularly flattening of the anterior, the head of the hippocampus. And again, just to show you again, this is an example of another case with late, with severe head of the hippocampus, hippocampal atrophy, involvement of the hippocampal body, and then this really significant thinning in the collateral sulcus. You can kind of see in more lateral temporal areas, thick cortex here. There's very little cortex, basically into the perirhinal cortex in this case, and widening of the sulcus. We've looked as well at sort of the pattern within the medial temporal lobe using more advanced imaging methods across the entire medial temporal lobe in a pointwise manner. And so we've looked at cases with Alzheimer's disease with or without TDP-43, and importantly, tau tends to correlate along the long axis of the hippocampus, whereas TDP-43 tends to be more associated with anterior structures. And actually using ratios of anterior to posterior hippocampal regions, we get reasonably good AUCs for discriminating AD cases with and without late. Another imaging biomarker that has been talked about is FDG PET, in which cases with hippocampal sclerosis tend to have really severe involvement in more medial temporal metabolism that's greater than what we see in typical Alzheimer's disease. And if you compare late to Alzheimer's disease, again, there's greater hypometabolism in cases with late, but less metabolism or hypometabolism in more inferior and lateral temporal areas. And a ratio of that also has been shown to discriminate cases with late from AD. And this is work that David Jones has done at Mayo. All right, but really the big challenge is when late co-occurs with Alzheimer's disease. As we've talked about, it's incredibly common as you get into later stages of Alzheimer's disease with moderate and severe CIRAD stage and Braak stage 5 and 6. At Braak stage 6 in this series, more than 50% of cases had concomitant late. It's worth pointing out, by the way, we think of like high ADNC as late a sort of late stage of Alzheimer's disease. Really, when people start to have clinical symptoms in Alzheimer's disease, they're kind of at an early part of high ADNC in many cases. And so this isn't just reflective, late is not just reflective of a very late phenomenon in the clinical features of the disease. Late with Alzheimer's is also associated with a more aggressive clinical course, much more aggressive than AD alone, and certainly late alone. This has also been shown looking at cases with Alzheimer's disease with and without late and hippocampal atrophy over time. In red, you can see there's greater atrophy or faster rate of atrophy in cases with TDP and concomitant late. So another case, this is a case where I think there's evidence that they had late and AD together. It's sort of an interesting, I think, an illustrative case. This is a woman who I saw in her late 70s who had primarily memory complaints, was forgetting appointments, conversations, decreased orientation to time. She had a mini mental of 28 out of 30. This is, again, our psychometric battery, just to confuse you now. The higher your level is, the more your percentile performance is relative to age and education adjusted norms. And so 1 or 1.5 standard deviations below the mean is reflective of some sort of threshold of impairment. And in this case, these are all the memory areas of measurement, and particularly delayed memory. And again, her performance was either borderline or impaired on memory measures. She did, though, show some degree of executive dysfunction, at least with clock drawing. She had low A beta on CSF, but a normal tau and phospho-tau. And given her low A beta, she was someone we diagnosed with mild cognitive impairment due to Alzheimer's disease. She did progress to dementia level impairment, or what we felt was dementia level, the next year or so. And we then said she had dementia due to Alzheimer's disease. This was her MRI. She has really small hippocampi, particularly the head of the hippocampus. I would argue you can see some thinning again in this, in the collateral sulcus relative to the right side, actually, is a good comparison. So more on the left than the right. She did have some degree of small vessel ischemic disease changes. And actually, if we quantify her hippocampal volume, in red here is the 75th, the sort of top dotted line is the 75th percentile, and the bottom line, the 25th percentile for typical Alzheimer's disease cases. And she was way below that. So she had really severe hippocampal atrophy. And after 15 years, so even though she progressed to dementia within a year or two of when we first saw her, it took her another 15 years to pass away. And by the time she passed away, she still was only intermediate Alzheimer's disease. So she was BROC 3-4 using traditional BROC staging. She was late stage 3, though, and she had amyloid angiopathy. And I think she's an interesting case because when we first saw her, she would have qualified for anti-amyloid therapies. But I guess one question that sticks in the back of my mind is that presentation was late. Was Alzheimer's which was driving her symptoms, or was it late? If she only was BROC 3-4 by the time she died, was she someone who early on, given how much hippocampal atrophy she had, was her symptoms really initially at least driven by late? I would argue probably so. And I think it's worth noting that about a third of cases with mild cognitive impairment that are amyloid positive also have evidence of hippocampal atrophy, but don't have evidence of tau. And this is some data from our group. And in fact, if you look at these cases, they show a pattern of atrophy relative to cognitively unimpaired individuals that's very late-like in isolated and more anterior medial temporal lobe structures. And so maybe a significant proportion of these cases have late as the driver of their symptoms. Well, I think that case maybe gives us a hint as to one thing that could be helpful in distinguishing cases with AD who have late versus not. And I should mention that that case is an example where maybe late was a sort of preceding pathology, but many cases, both pathologies are likely occurring simultaneously. And if we go back and look at the postmortem hippocampal volumes that I mentioned earlier and compare cases with Alzheimer's disease versus cases with Alzheimer's disease and late, the cases with both diseases, again, not surprisingly, have much more severe hippocampal atrophy. And if you kind of, in your mind's eye, take the bottom quartile of the range of Alzheimer's disease, that bottom quartile of atrophy is really enriched in cases with late. And so I think a lot of what we think about in the range of Alzheimer's disease historically, the bottom part of the range in terms of memory and hippocampal volume are likely cases driven by late. We took that cutoff. We just defined a bottom quartile of cutoff in Alzheimer's disease to say in cases with amyloid pathology, how enriched are those cases in cases with late? And from the Alzheimer's disease neuroimaging cohort, we found if we applied that bottom quartile cutoff, it was highly enriched in late. Almost 85%, or around 85% of individuals had concomitant late. Whereas if we looked at the median and above, only about 30% had concomitant late. We then applied, and I won't get into the details of this, something called a SILA model, which is a way of estimating tau onset based on tau biomarkers. And so we can kind of place people on their tau age. And if we look in the cases that are above the median, cases we would suspect largely just have late, they show a pattern on memory that's what we expect with Alzheimer's, where they don't really change until they're tau positive, because we think tau is what drives symptoms. Whereas if we look at the cases that we think are mixed, we actually see changes before they become tau positive, suggesting a second process may be driving memory symptoms. More broadly, we have been very interested in trying to think, how can we, in an individual patient, get back to the idea of figuring out how much of their symptoms are driven by Alzheimer's disease versus other processes, including late. And I've alluded to this already, but tau is actually a pretty good metric of telling us what stage someone's at with regard to Alzheimer's disease. In fact, it's even incorporated in the US, the Alzheimer's Association's new clinical criteria. And I think this is nicely illustrated in a paper by Bill Jagis's group, where they show tau uptake based on, or tau PET uptake based on fluorotaucipir imaging at baseline in patients with Alzheimer's disease, hotter colors being more tau. And it just beautifully matches where you see longitudinal atrophy over time in those cases. So tau is really heavily linked to atrophy. And so we kind of thought, well, maybe tau tracer distribution and intensity can kind of be an answer sheet for what we would expect the pattern of atrophy and degree of neurodegeneration to be due to Alzheimer's disease relative to other conditions. And so we have tried to create these kind of normative relationships between tau and atrophy, in this case, cortical thickness, across different brain regions. And what we see, this is right inferior temporal gyrus, y-axis is thickness, x-axis is tau PET uptake in that region. We see an expected linear relationship, the more tau, the greater thinning. But we also see cases that have less atrophy than expected for tau, and cases that have more atrophy than expected for tau. And we expect those cases, at least with more atrophy than expected for tau, maybe they have a copathology driving that greater atrophy than you expect. And so what we can do is across the entire brain, we can create residual maps of atrophy once we've kind of removed the effect of Alzheimer's disease. And actually we can cluster these individuals. We see different spatial patterns. We have a canonical group, the kind of Goldilocks group, where there's the right amount of atrophy for a given level of tau. But we also see a limbic vulnerable group and a diffuse vulnerable group, and then also resilient groups as well. But that limbic vulnerable group looks quite a bit like people who have late, and so that's what we'll focus on here. And I think importantly, these groupings also tell us beyond tau, so controlling for tau levels, about what happens to these patients over time. And so black is the sort of canonical group, and in orange is that limbic vulnerable group that has a much faster rate of atrophy, or greater rate of cognitive and functional decline over time than the canonical group. And actually the resilient groups, if anything, show very little change over time. Well, we felt that if our residualized maps reflect other factors or pathologies that are driving atrophy or resistance to it beyond the case of Alzheimer's disease, well, then we should see in amyloid negative symptomatic cases, if we just look at their patterns of atrophy, they should kind of match these patterns as they'll reflect those other processes. And that's exactly what we did see in amyloid negative individuals. We saw a limbic predominant group in particular that matched the residualized limbic predominant group. And in fact, those maps look quite a bit like the maps we saw in postmortem imaging. And in fact, actually, the heat maps, in our case, orange is more severe and yellow is more severe in the postmortem example. So they're kind of in opposite directions. But there's incredible overlap both in distribution and severity. So it does seem to suggest maybe late is what's driving these groups. We've created this limbic vulnerable group in a sample that we had postmortem data for and antemortem imaging, and again found this limbic predominant group. And this group indeed was associated with higher levels of TDP-43 in limbic regions than the canonical group. And actually, if anything, in general, and this is true in our resilient groups beyond just the limbic group, there generally is less pathology in the resilient group. So part of being resilient is not having co-pathologies. To sort of further validate this approach, we've also looked in postmortem imaging. So we scan all of our patients who come to autopsy at our center in at least one hemisphere. And we're able to do really granular measurements of the medial temporal lobe, both histologically and in terms of imaging to match up histology to imaging. And what we see is we created a regression model in CA1 of quantitative measures of tau pathology in patients on the AD continuum versus volume on the y-axis, just based on cases who have tau alone or AD alone. And so that kind of gives us the true kind of normative map, what you really want to get. And then if we plug in cases with concomitant late or late alone, they have negative residuals quite significantly. And we can actually look across the medial temporal lobe in a pointwise manner and beautifully in red, the hotter colors. The areas that have negative residuals are CA1 and subiculum, where we expect to see TDP-43, and entorhinal cortex moving into the collateral sulcus. So kind of fitting with what we've been describing. We have looked more focally within the medial temporal lobe in the in vivo brain, using global measures of tau or local measures of tau as well. And in this case, actually, we're using p-tau217, which, while it's very sensitive to amyloid, it also, to a large extent, actually does correlate with global measures of tau pathology. And so I'm not going to go into detail here, but we also could create a vulnerable group that we were able to cluster that has a pattern very late-like in greater atrophy than expected for a given level of tau. These are more anterior regions of extrahippocampal medial temporal lobe. So this is entorhinal cortex into the collateral sulcus, as well as significant involvement of the hippocampus, particularly in the head, but really the whole body. And if we look cross-sectionally and longitudinally, we see this more anterior temporal polar pattern of atrophy, again, controlling for the degree of tau pathology, supporting the idea of concomitant late pathology. And much like what we did before, if we look at tau onset age, in this vulnerable group, we see greater change in function and in memory prior to the onset of tau pathology relative to the canonical group in black, and even slower in the resilient group in blue. And the important point is, if we use p-tau217, which we did here, and structural imaging with T1 MRI, we can apply these methods to patients that are coming into our cohort who are receiving anti-amyloid therapy. And we are able to find that about a third of them are actually in this vulnerable group, and they look like late in terms of their pattern of atrophy, whereas less than half are in a canonical group, and we have some resilient as well. And so what we're trying to do at our center is classify patients, and we're going to look at their outcomes and see the degree to which these, at least markers of possible co-pathologies, influence those outcomes. I'm going to actually skip this just for the sake of time, but we've also been doing this in a more global way, looking at measures of tau and cognition with just plasma p-tau217 and simple cognitive measures, and are also able to define these resilient and vulnerable groups that map onto late, but also alpha-synuclein pathology. But I will save that for another time if you all want to talk about it, because I wanted to end just on what our clinical criteria are for late. I think I provided a kind of background to at least motivate how we thought about giving an initial attempt at clinical criteria. And so these criteria that we published about a year ago were based on a committee of 36 experts across the world who met at an NIA workshop that I co-chaired with Julie Schneider in 2023 to discuss gaps and opportunities in late. And I think part of that discussion led us to thinking, well, maybe it's time to kind of put a flag in the ground as to what might be reasonable clinical criteria that we could try to validate and then iterate on. And so we did that. We realized that in the absence of a specific TDP-43 biomarker, diagnosis is going to be at best probabilistic. But guess what? That's what our field's done for years. We talked about probable and possible AD prior to our biomarkers for that. And I think other neurodegenerative conditions like FTLD is certainly probabilistic as well. We defined two contexts. One, when late was the primary driver of symptoms without significant ADNC. And for those patients, we can call possible or probable late. Whereas we also defined a pathway for saying someone has late NC mixed with Alzheimer's disease neuropathic change. And we felt at best we could be tentative and say possible late in those cases. We defined a clinical syndrome for both of them. In the late predominant cases, we'd expect, as you might think, based on what we've described, a primarily amnestic syndrome that is temporalimbic in nature with other cognitive domains largely spared. Generally, these patients will be over than 75 and will have a pretty indolent course. Whereas the core clinical syndrome for AD with late is much vaguer because we expect to see a progressive amnestic multi-domain syndrome, which is what we see with Alzheimer's generally, but where memory is more severe relative to other domains and that the course tends to be more rapid. So taking that, those syndromes, if people have the core clinical features of late as a primary driver, they need to have hippocampal atrophy. I think none of us feel confident we can make this diagnosis without really significant hippocampal atrophy, particularly Shelton's 3-4. If you have no amyloid biomarkers available, I think the best you can say is possible late because many of these cases probably do have Alzheimer's disease. But if you do have amyloid biomarkers and you're amyloid negative and have that degree of atrophy in that pattern, I think we can be fairly confident that the vast majority of those cases are going to have late. If you're amyloid positive, it doesn't mean that you can't have late, as that one case I mentioned earlier illustrated, but you really need to be tau negative, and really tau PET is probably the best measure for that. And there we said that you could have possible late, partly because there's some disagreement about how sensitive tau PET is to early tau pathology that can drive atrophy and symptoms. If you're tau positive, though, you have Alzheimer's. You have amyloid and tau pathology symptoms and hippocampal atrophy, but you still could have concomitant late. So that takes us to the other side of the other sort of syndrome. And in these cases, you really need other supportive features like severe hippocampal atrophy out of proportion to mild dementia or out of proportion to the degree of tau pathology, as we've talked about. That FDG PET signature we talked about also can be helpful in these cases. So to conclude, late is common and an important driver of late life cognitive symptoms. It's common co-pathology and modulator of Alzheimer's. And there are imaging and cognitive features that I do think reflect the distribution of TDP-43, and that can help us in a probabilistic designation of late that I think is a reasonable thing for us to start to classify in vivo in our patients and follow those patients hopefully to autopsy. The clinical criteria really do require validation in large in vivo studies. So this is a sort of consensus-driven idea, but we need much more data in vivo. So just want to thank a variety of my collaborators. Underlined are people who did some of the work that I presented today. And then also just want to thank the collaborators who helped with developing the clinical criteria, and really want to thank all of you for your attention today.
Thank you, David. Wonderful tour de force. You're doing pioneering work, and we're glad to be watching. Maybe if you could take your share down, we can go to questions. I see Ioannis has a question.
There we go.
Actually, I have two, but first of all, I want to convey the apologies of Ioannis Sotiropoulos, because there was a flood in his basement and he had to work with this. But I'm sure he's very interested and he's going to see the recording. So thank you for this great presentation and on this very interesting topic. Actually, this is something that I think is universal. We have also a small brain bank here in Crete, and in 10 brains so far that we have proven that they have Alzheimer's disease. Of course, they are advanced stages, so they are BRCA6 or BRCA5, most of them, and they have FAAN5 in most cases. So they have in 9 out of the 10 brains we have examined so far, they have TDP43 in the hippocampus. So I think this is universal. Of course, maybe the samples we have are over 80 years old. So first of all, do you think because this is mostly because of the advanced stage or the advanced, let's say, BRCA stage they have?
Yeah, so I think both are very linked to a greater enrichment in people who are likely to have underlying late. So certainly age influences the presence of late and the presence of late as a co-pathology with Alzheimer's disease. So, there's data, very nice data in early onset Alzheimer's disease relative to late onset disease, where, for example, alpha-synuclein pathology, which tends to occur also as a relatively common co-pathology, that there's actually not that much of a difference between age and Alzheimer's disease in those cases. So in both cases is around 50% of the cases. Late is much lower in these early onset Alzheimer's disease cases relative to late onset Alzheimer's disease. And so age also does modulate. They're not, by the way, just to point out, they're not completely absent though from these early onset cases. About 10 or 15% also will have late there. Why you all have 90% and it's a little hard. It's a little hard to know. Obviously, the older your sample gets, we again in our over 200 cases ended up somewhere around. It was, I think, around 40% had late stage 1 through 3. It'd be interesting if you continue to see those high numbers, and if we can kind of think about what are the reasons why in your population you're seeing this higher rate.
That's very interesting. Of course, it's 10 brains, so 9 out of 10, but still, it's a great surprise to us that we have so many TDP43, let's say, deposition. A second question, if I may, Ioannis, the other thing we found in our population is that in Alzheimer's patients phenotype, so we have Alzheimer patients diagnosed by the criteria for Alzheimer's disease. In 2 of these patients, we found the one pathogenic variant in the TDP43 gene, which is the TARDBP. So 2 old people, 85 years old, they match the criteria for Alzheimer's disease, and still we found the variant that has been described so far to cause ALS and frontotemporal dementia. So do you think there's possibility that in some of these cases that we call late, there is maybe a mutation or a variant that's somatic and that starts the pathology inside the brain and they manifest as late?
Yeah, so I think it's a really interesting question, because I do think if you look at there are some small sort of GWAS studies with late that have been done. There is some overlap between the risk alleles for FTLD and ALS and late. So like TMEM106B and the granulin, there's a granulin SNP that's also associated with late as well. And so there is certainly some shared genetics between FTLD and late. Obviously, there's a shared pathology in those cases as well. Why someone would present with FTLD versus late is sort of hard to know. I mean, there's been a little bit of data that that cases with FTLD mutations, autosomal dominant mutations, if they present later in life, tend to have a little more medial temporal pathology than earlier in life, kind of akin to like what we see with Alzheimer's, where you tend to have more cortical involvement in the younger onset cases and more limbic involvement in later cases. So maybe there's some interaction with age, but I think we don't really know. And there are definitely boundary cases as well between late and FTLD, where we've had a few cases where, they have an amnestic presentation, but there's spread of TDP-43 that, really kind of overlaps with what might be an FTLD case. And actually the networks that are involved in late really overlap with semantic dementia, which tends to have TDP-43 as its primary pathology. But interestingly, the TDP-43 pathologic type is very different looking in a semantic dementia than it is from late TDP type. It's sort of TDP-43 type C is what's associated with semantic dementia, where late looks more like TDP-43 type A. And so again, there's these overlaps, and it's interesting in terms of the regions that are affected, but why someone moves in one direction versus the other is I think, really important work to do.
It's 11 o'clock here on the East Coast. David, can you stick around for a couple more questions?
Yeah, I have a couple more minutes.
Yeah, let's go to Nick.
Well, hi. So it's really great presentation because you summarized everything so nicely, and you remind us many things, and we learned a lot from this. So can I get your help in a clinical case? So
yep.
Yeah, so I think very borderline. So she's 75. Actually, she is the mother of a physician. He's a nephrologist in Britain, and over the last few years, she has been forgetting. So her mini mental is 25. And her profile is amnestic, both verbal memory and non-verbal or visual memory. The other domains are pretty much preserved. And functionally, she's in between MCI and dementia. Okay, and so her MRI has a medial temporal atrophy, quite prominent. So maybe even more than we see in AD. I have to admit that I didn't look at the anterior to posterior ratio of the atrophy. And she's actually epsilon 3. Some family history of dementia, but at an older age, and she's 75. So I said, okay, in the beginning, I thought it looks like Alzheimer's disease. We got it, a p-tau 217, and it was borderline. Then we get an amyloid PET, and it's read as positive. But talking to the nuclear medicine physician, they say, it's borderline. It's like about 25 to 30 centiloids. So yeah, we called it positive, but it's not really, mind-blowing. So there we are stuck because of the possibility of giving donanemab.
Yep.
In her case. So I thought, as a next step, maybe, to get an FDG PET and look at the ratio, medial temporal to frontal or medial temporal to more inferior temporal, and possibly get a tau PET. Thinking that, okay, if tau PET is negative, then maybe we have amyloidosis, but it hasn't translated yet into neurodegeneration. So we should attribute her amnestic symptoms more to late type rather than. So, I mean, I don't know, just because we need to make a decision on her, do you have any other inputs or anything else to look into that could help us, make the correct decision for her?
Yeah, no, it's funny. These cases come up not infrequently. We have a case conference on Monday for decisions about anti-amyloid therapy. And, we really struggle with these cases. Actually, I showed a slide for a paper that hopefully will come out fairly soon in Brain, where we are looking at the relationship between plasma p-tau 217 using the Lumipulse platform and level of atrophy. And that your case, if they have relatively severe atrophy but are borderline on Lumipulse, likely would fall into that vulnerable group that we would think of as being much more likely to have late. And, with Lumipulse, at least, we've been looking a lot to try to define tau positivity based on its values. And, the cases, we really don't start to see significant tau signal till cases are again, it's only on the Lumipulse scale, but like 0.3 or above. And so when we see these intermediate cases between what they call positive and that 0.3 level, again, it's very uncommon for you to have a lot of downstream atrophy, and it's also uncommon to have a positive tau PET. I think tau PET in these cases is probably the most useful measure, because if it's tau PET negative, then I think it's pretty unlikely in my mind that you have enough tau in your medial temporal lobe to cause really significant atrophy if it's not visible on the tau PET scan. And so in those cases, I would think, late is more likely the cause. And I think if you can get it, it's probably the most helpful in this context.
Okay, great. Thanks. It's very helpful.
But we struggle with those cases are so hard.
Yeah.
Other questions?
Can I ask a question? It's a very practical question. Thank you for the great talk. We work in iPS cells and generating organoids from Alzheimer's disease patients. So usually we take a little bit, we are using a little bit of blood of Alzheimer's disease patients, and we isolate peripheral blood monocytes, we generate their iPS. So we have a high interest in generating these from late patients. Do you have any blood stored from these patients? We talk more, but I wanted to ask now.
Yeah, I mean, well, we have plasma stored, so that's the primary biofluid that we have in those patients. So you need like PBMC,
Yes.
yeah. So we don't. There are other groups that do, and like through NECRAD, which is connected to the National Alzheimer's Coordinating Center, there are definitely cases in there that I bet you have autopsy data and cells available that I would probably look into that. We certainly could do that in the future also. I also, Kostas, you all collect, you guys are doing iPSCs as well at Hopkins.
Well, Basso is doing our iPSC.
Yes,
I'm
doing there.
That's right. That's right. Yeah.
Yeah, but because they haven't started collecting the blood yet, I thought maybe, we could move a little bit faster. And if you had some blood, we could work together. That would be great.
Yeah. No, we have plasma, but we have not. That's all we've collected. But I'd be very interested in thinking about, moving forward if you want to collaborate or think about that as well. But I do think
that would be great.
I think NECRAD probably does, though, have some opportunity to collect those cells as well.
Perfect. Yes, we could talk more. Thank you.
So we could reach out to Tatiana Farud, who runs NECRAD, to ask her if there's a way to find out. That's probably the next step, where they have a diagnosis.
Yeah, and David has a needers to work together as well, so that would be great.
Oh.
Perfect. Thank you so much.
Yeah, no problem.
David, I will say every time I hear you speak, I learn something a whole lot more interesting. So you hit it out of the park today. We're grateful that you participated in this. This is, as you know, recorded, so lots and lots of people hopefully will have a chance to benefit from this. So thank you for being part of the seminar. And
thank you very much. It's very, very kind of you. And I really enjoyed the conversation.
Excellent. Okay, so we will go ahead and adjourn the seminar. Take care, everybody.
Bye, everyone.